So in the last video, I focused on alpha helices, and that was one of our three major secondary structural elements. And that terminology, secondary structure elements or secondary structures, I want you to think about those as kind of interchangeable. Now I'm going to talk about our second, which is our beta sheets. These are kind of known as the extended conformation because they generally use more than 10 amino acids, which was the average size for an alpha helix. So in a beta sheet, you have backbone hydrogen bonds of neighboring polypeptide chains, interstrand, not intrastrand, in alpha helix. So we have strands that look something like this. So remember that what we're looking at basically is a directionality from N to C. So this blue corresponds to a nitrogen, C, C, N, C, C, N, C, C, and so on. We have a direction going in this way, and then in the opposite end, we have them going in this direction. So this is referred to as an anti-parallel strand or an anti-parallel beta sheet. Now, what I really want you to take away from this is let's look at these two different formations. We have an anti-parallel and a parallel. Now, between those two, which one do you think is stronger? If you said that it's the parallel because, well, it's not anti, that would be incorrect. The correct answer is our anti-parallel beta sheets are stronger than the parallel beta sheets. And the reason for that is, well, let's look at the distance. Just look at a relative measure here, that versus this. And think back to Gen Chem 1, strength of bonds. Shorter bonds are stronger than longer bonds. And one of the reasons that these anti-parallel beta sheets are stronger is because these hydrogen bonds are shorter and the atoms involved in these hydrogen bonds are in one straight plane, which is something that we previously talked about when we talked about hydrogen bonding. So here's some different models for parallel and anti-parallel beta sheets, as well as a mixed beta sheet where you have a combination of parallel and anti-parallel. Now, like I said, which types are stronger? It's going to be your anti-parallel. Now, what is the orientation of the donor and acceptor in a strong hydrogen bond? Well, orientation puts them directly across from one another. Now, these beta sheets, they can kind of organize and they can organize and make more complicated structures. For instance, large beta sheets, there are structures which exhibit a right-hand twist. Now, what I want you to see whenever you look at this model right here is we have basically one polypeptide. And you've got some alpha helices. And then you've got these arrows, which are beta sheets. The connections between the strands can either be simple or they can be complicated. Now, to look at and visualize a parallel versus an anti-parallel, this would be your parallel. This would be your anti-parallel. And it's hydrogen bonds that are stabilizing this. Now, what about the amino acid side chain? So what about those R groups? Well, the side chain should cover and protect the backbone hydrogen bonds that are going to lead to the formation of the secondary structures, whether it's an alpha helix or a beta sheet. Now, what that's going to lead to is in this model C right here, the R groups, the amino acid R groups, are going to be protruding outward from the helix. Now, what that means is those R groups can form ionic interactions, which make up your tertiary structure with any of the things in their surroundings. Now, a fair question here is, okay, if that's what an alpha helix looks like, what are some amino acids that are going to lead to formation of an alpha helix? They have a size preference with respect to those different amino acids. The coil of an alpha helix constrains space between adjacent and vertical amino acids. So the preference is for smaller and less bulky amino acids. So extended and non-beta branched. If you remember, you have, whenever you're looking at a polypeptide, you have your alpha carbon and then another carbon. This is your beta carbon. So no branching off of here. So good amino acids would be something like let's say like a aspartic acid, maybe, because that's just got 3 carbons or 2 carbons off of it. But threonine, valine, isoleucine, those are all good candidates. Amino acids that form favorable or that form ionic interactions: alanine, glutamic acid, methionine, leucine, phenylalanine, glutamine, histidine, lysine, arginine are all good candidates. What I want you to take away from this is an alpha helix, what are the desired properties of an amino acid and whether or not they're going to be found in an alpha helix. So which amino acids or which properties are favorable for the formation of an alpha helix. So that's kind of where I want you to go with that. Beta sheets, the extended structure of a beta sheet leaves the maximum space for amino acid side chains. So that means large, bulky side chains prefer to form beta sheet structures. So tyrosine, tryptophan, phenylalanine, methionine, those are great. And bulky and awkward beta branched amino acids are also good candidates, which I'm sorry, on the previous slide, these are all your beta branch, so bad for alpha helix. That would be your isoleucine, valine, and threonine. Those are great for beta sheets, however. Cysteine is also a good one for a beta sheet. So those amino acid side chains are absolutely relevant to are you going to form an alpha helix or a beta sheet? Now, both sheets and helices can be amphipathic and amphiphilic. So you could have one portion that's a bunch of polar residues, and then a portion that's non-polar or more hydrophobic residues. What that means is you're going to have, well, this region right here that would love to interact with other non-polar portions or non-polar substances. So when it comes to the organization of an alpha helix, it is important to know which is going to, when we're, talking about the helical wheel, amino acid 1 is in this position, amino acid 2 is in this position, 3 is in this, 4 is in this, 5 is in this, 6 is in this, 7 is this, 8 is this, 9 is this. That is important because it gives you an idea of, okay, what side of this helix is this amino acid R group going to be found? So that's where that those different positions are really important, because if we look, this question at the bottom, what can you say about the location of 1, 4, 8, and 11? Well, 4, 8, 11, 1, right there. These are amino acids that in a straight polypeptide, so looking at the primary structure, they're far away from one another, or they're separated by like 3 amino acids each. But when you form that secondary structure, they are on the exact same face of that alpha helix. Secondary structure breakers, so there are some amino acids that have nothing to do. There are some amino acids that, so just as we have some amino acids that have a preference for forming an alpha helix or a beta sheet, there are some amino acids that will disrupt the formation of an alpha helix or a beta sheet. Glycine in particular, if you think about the R group for glycine, simply a hydrogen. Glycine is considered to be too flexible. The side chain hydrogen is too small to protect any sort of backbone, so it's going to disrupt any sort of backbone formation. Proline has the opposite problem of glycine. Proline is extremely rigid, and it restricts the phi angles to -60. So proline is never found in the center of an alpha helix. It will disrupt an alpha helix. So there are also, those two amino acids, glycine and proline, they kind of capture opposite ends of the spectrum and equally present problems. Strong hydrogen bonding side chains compete directly with backbone hydrogen bonds. So asparagine, serine, aspartate are all good hydrogen bond formers. But one thing to keep in mind is, what is aspartate more likely to form? Ionic interaction. Clusters of breakers. So if you have multiple of these breakers kind of packed together, then you're going to give rise to our third form of a secondary structure, and that would be a beta turn. Sorry, I just had to eat a chip. These mark the boundaries of alpha helices and beta sheets and serve to link up secondary structure elements. Tight turns consist of, or our beta turns consist of 4 to 5 amino acids in a well-defined structure. It's not completely repetitive like an alpha helix or a beta sheet, but this is a distinct set of amino acids. So a beta turn, there are 2 different types of beta turns. There's a type 1 and a type 2. And in a type 2, there will always be a glycine, which, if you remember, glycine is a secondary structure breaker, but it's also kind of a secondary structure former because it breaks a beta sheet and leads to the formation of a beta turn. Proline and glycine are both very common in beta turns. So they break, you can kind of look at it as they break secondary structures and give rise to a turn, which is also a secondary structure. 10% of the proline residues in proteins follow a cis peptide bond. Now, different amino acids have different scorings different amino acids have different propensities to be found in alpha helices and beta sheets. And in the 1970s, Chow and Fastman were 2 researchers who said, okay, we're going to look at all the secondary structural knowledge that's out there, and we're going to try to score every amino acid and give them a propensity score for, are they likely to be found in an alpha helix or a beta sheet? And what they did, and what continues to kind of be the gold standard to date, is to take a primary structure and basically use these scores to come up with the likelihood of an alpha helix or a beta sheet. So the scoring system ranges from 0 to 2. And if you look at our glycine, has a score of 0.57 for a propensity for alpha helix and a 0.75 for a propensity for a beta sheet. Now, the value of this is it really gives you a way to go from primary, or predict secondary structure based on the primary sequence. Up until this was done, people looked at protein structures and had some rough ideas, but they really consolidated everything to date into, great, now we can predict what a secondary, or what secondary structure element forms, if one forms at all. This is very powerful, and they came up with, based on those scores, a couple of ideas. The proposed final secondary structure is the consensus of all amino acids within a region. So a minimum of 4 of 6 amino acids should show an alpha helix preference in order to form an alpha helix. 3 out of 5 amino acids should show beta preference in order to show a beta sheet formation. Clusters of 2 to 3 breakers in a sequence of 4 are needed to set the secondary structure in any region. And individual misfits adopt a secondary structure of their neighbors. So if there was a single, let's say glycine, which is generally too flexible, if there's a single out of like 60 amino acids that form an alpha helix, that alpha helix is still going to form. However, they came up with these kind of guidelines and ideas. And then as a kind of further example, if you have 1 or 2 helix preferring amino acids scattered in a majority of beta sheet formers, the structure adopted is entirely a beta sheet. Proline is never found at the center of an alpha helix, kind of as a, yes, this is a misfit, but it's a misfit of misfits. It's a really bad one. It's a bad egg. Okay. Now, this is very powerful to be able to go from primary structure to secondary structure. It is absolutely invaluable because ultimately, if you know organization of biological molecules, you get a little bit of a sense of what it does. And so if you can go from primary structure to quaternary structure, that's huge. And so that's still something that's being actively researched, but to go from primary to secondary is big, from secondary to tertiary is huge. And so that's kind of where the direction that is being, where it's going. Now, the importance of these different levels. Well, sickle cell anemia, whether or not your blood cells, your red blood cells form these sickle shapes or these wild type shapes, is dictated solely because of a single amino acid mutation in hemoglobin. Whether or not your red blood cells will look like this or this is because of one amino acid. There are tons of diseases that have that same sort of thing. So protein sequencing is very important. Getting the primary structure of the protein is very important. Sequences and composition reflect the function of the protein. Membrane proteins have more hydrophobic residues, whereas fibrous proteins have atypical sequences. Homologous proteins from different organisms have homologous sequences. As an example, primary sequence of a protein is species dependent, but if you compare protein sequences or primary sequences of homologous proteins to determine which residues are important for function or for structure and function, we want to find, whenever we're looking at protein sequences, what are some invariant residues or conserved residues? What are the residues that are the most important? Then we also want to look at, when we're comparing sequences, what are some conservative substitutions? For instance, if in hemoglobin alpha, let's say we have an aspartate in one organism and then a glutamate in another organism. Well, the properties of these two amino acids, well, they're very similar to one another. The R groups look effectively like this. Or I mean, that's what those two protein R groups look like. So they're very similar to one another. But then also in protein sequences, you want to find the hypervariable regions. Are there regions of these proteins where there's huge differences? So here's how we look at different protein sequences and organize them. Amino acid sequences of cytochrome C from 38 different species. Our aim whenever you do something like this is to basically find what are the amino acids that do not change across every single organism, and what, because ideally these proteins all do the same thing. And then are there some mutations where it might be an alanine in one organism and a valine in another organism? That's what we kind of want to do, so that we can take that information and develop kind of an evolutionary or a phylogenetic tree based on protein sequences. Anytime we do large scale sequence analysis, there's tons of different resources that you can use. All of these are not completely out of date, but there's still fundamentals that are being evaluated, that are being looked at with these. There are tons of different data banks that contain protein sequences. XPASI is a common one. The XPASI molecular biology server. These are all great for comparing protein sequences. Now, protein sequencing. This is a technique, or this is something that's valuable, but I'm going to ask you questions about the chemicals and what they do, but not about reassembling these. I think that there's some similarities between protein sequencing and restriction endonucleases. But first and foremost, whenever you're doing protein sequencing, you got to determine the total amino acid composition of the protein. And what you do is you completely hydrolyze that protein. You break it up with 6 molar HCl at 100 degrees Celsius. You determine the order of the amino acids in a chain, and the method of choice for protein sequencing. Sometimes it's Edmond degradation, and what you're going to do is you're going to remove one amino acid at a time from the N termini. You're going to remove an amino acid, identify what that is. And basically you do that over and over and over again. This can be automated, and it works with peptides up to 50 amino acids long. The downside of this technique is that it does not provide 100% yield. You must cleave a protein into smaller sizes. If you're working with something that's like 800 amino acids, you've got to break that thing that's 800 amino acids into 50 amino acid bits. Disulfide bonds, remember what we talked about previously with cysteine or 2 cysteines forming a cysteine. Well, that is something that you've got to work with that, because that's not simply something that you can adjust the pH, and that covalent disulfide bond is going to be broken. So you have to break that disulfide bond. Now, the steps in sequencing: first, you got to separate any multimeric protein into individual subunits. Next, you got to break the disulfide bonds. Third, determine the amino acid composition. Fourth, do N group analysis. Fifth, use molecular scissors. Sixth, repeat step 5 with different molecular scissors. And 7, sequence each purified peptide fragment with Edmond degradation. And finally, reconstruct that sequence. So there's a lot to consume with this, but here's a great visual, in my opinion. Now, what this shows is a diamond. We've got 2 polypeptides. One's blue, one's green. Those polypeptides are held together with a disulfide bond. What you're going to do is reduce the disulfide bond and separate the chains from one another. So as soon as you've separated these 2 polypeptides by breaking the disulfide bond that exists between the 2 cysteine residues, you're going to use chemical methods to break them down. Chemical or enzymatic methods to break them down. And then you're going to determine the sequence of each of these individual peptides. Now, the value of this, or the importance of this, is that you're basically using enzymes or chemicals that cut after specific locations. So these are known as your molecular scissors. Now, what I'm going to do is we're going to get into some examples, and I'm going to walk you through some of those. Now, here's the comprehensive 8-step strategy. Use extreme pH and urea, or 8 molar urea, or 6 molar guanidine hydrochloride. These extreme conditions that are going to disrupt basically all types of interactions. Then you're going to, and this is very important, these things are very important. I want you to know what DTT is, dithiothiotol, and 2-mercaptoethanol. These are substances that break a disulfide bond. So DTT and 2-mercaptoethanol. Those 2 substances I want you to know. I'm telling you explicitly, I want you to know those. The remainder of these, I'm not too concerned about, because we're going to be focusing primarily on the molecular scissors. So you cleave the disulfide bond, determine the amino acid composition by HPLC. So that tells you, yes, every single one of our 20 protein-genic amino acids is present. Okay, valuable. Then this other technique, or the step 4, is N group determination, where you're going to use dansyl chloride, FDNB, or similar reagents to determine what's the N-terminal amino acid. And then these other carboxypeptidase or exopeptidases will cut off the C-terminal amino acid to say this is the amino acid that's on the C-terminus. Now, you're going to remove a couple from each end at a time, determine what they are, and go from there. Here's where I want to focus, though. These are molecular scissors. I want you to know what all of these molecular scissors do. I'm going to first focus in on trypsin, chymotrypsin, and elastase. Now, what these are going to do is they are going to look for Well, here, let's start at the top, or let's reverse back, and here's our top. So this is an alpha carbon, and this R sub n minus 1 tells you, or that would be a specific amino acid. Then here is your peptide bond that is going to be cut by one of these molecular scissors. It's a question of, is it going to be, what is the amino acid that has to be here and what has to be here? Okay. Trypsin, chymotrypsin, and an elastase. Trypsin is going to look to see if this R sub n minus 1 is an arginine or a lysine. Those are positively charged amino acids. If an arginine or a lysine is in this specific location, it is going to cut this bond. However, if in this position right here, we have a P or a proline, so if this is RP, then trypsin's not going to cut it. If this is RL, trypsin will cut that peptide bond there. If this is a lysine, followed by a leucine, trypsin is going to cut that. Because this specificity tells us R sub n minus 1 needs to be an arginine or a lysine. R sub n cannot be a proline. Now, next up, chymotrypsin. Chymotrypsin is going to look to say, well, what is in this R sub n minus 1 position? Is it a phenylalanine, a tryptophan, or a tyrosine? Well, then I want to cut it, unless there's a proline. So if you look at these rules, proline presents a problem. For trypsin, chymotrypsin, and elastase, if they see proline in this R sub n minus 1 position, regardless of what's in the R sub n minus 1 position, those enzymes are not going to cut this bond right here. Now, I want you to actually focus in on these three, the rules for these three. The others, I'm not going to ask you about those. But those three right off the top, those are the ones that I'm curious about. So the way that you can expect this in a practice problem would be, and I'm going to show you one practice problem in just a moment. And then there's another one at the end of the PowerPoint that you can do on your own accord. And it is a complicated one. So if you don't get it, but if you get the one that we did, or that I'll do, then consider that good to go. Okay, so if you were to see a polypeptide, something like F, let's go with M. That's a phenylalanine in the R sub n minus 1 position, and a methionine in the R sub n. Now, the question is, does trypsin cut this? The answer to that question is no. The question is, does elastase cut that bond right there? And the answer is no, elastase does not cut between F and M. Chymotrypsin, on the other hand, well, chymotrypsin is looking for this positions, n minus 1, to be a phenylalanine, a tryptophan, or a tyrosine. As long as chymotrypsin finds that, and then in the R sub n position, there is not a proline, chymotrypsin is going to cut that. So I want you to know trypsin, chymotrypsin, and elastase. Don't worry about cyanogen bromide. Cyanogen bromide, though, just for future, it cuts after methionine. But at any rate. So you'd repeat that. You'd do it with one, and then you'd cut with another, and then you'd cut with another, and you get a lot of information from that. Then you'd sequence all those peptides, and you'd reassemble it. Now, here's where I wanted to go. Here is a practice problem that I love. So this is demonstrating the steps being put back together. I'm going to scribble out some of this information so I could actually use. Okay. Okay. So this right here shows that we've got a polypeptide. The polypeptide has a disulfide bond. when you use dithiothiazole or DTT or beta-mercaptoethanol to break that disulfide bond. Then you use high heat and low pH. You can hydrolyze that polypeptide, and then you do an analysis and you find out, oh, well, there's 8 alanines, 2 cysteines, 2 lysines, 2 leucines, and 2 tyrosines, as well as everything else. There's tons of, like, there are a total of, let's see, this would be 6 plus 6, that's 12. And then let's see, that's 5 more. So we've got 17 of the total, or there are 17 unique amino acids in this polypeptide. Okay, great. That's valuable information that we got from this. Now, we have a grand total of 38 amino acids. And what we are going to do is we are going to do some analysis of this. And let me go ahead and draw some more boxes. So what we find is that, just a second. What we find, this first bit of information that we get, is that when we take this polypeptide, we break the disulfide bond, and then we give it one of these molecular scissors. We give it trypsin. We want to jump back for a second, actually. We want to say, what does trypsin cut after? Trypsin is going to cut after a positively charged residue unless there's a proline. And we are using trypsin because when we look at our composition of amino acids, do we have any positively charged residues? Yep, we got a lysine, we've got an arginine. Okay, so we've got those 2 residues. So what that means is trypsin is going to see, if it sees something like RA, it'll cut there. Okay, now what we get from the molecular scissors or using trypsin on this polypeptide is these 4 unique polypeptides. Now, the great thing about this is we know that trypsin is going to cut after an arginine or a lysine. How can we use that information? Well, we can use that information because we know trypsin is going to cut after those. We know that of these 4 different polypeptides, we know which one is the very end. That'll be this one right here. We know that's the end because if we look at the other 3, this ends with lysine, this one ends with arginine, this ends with lysine. We can use the information that we have as well as the knowledge that we have about trypsin and say, well, because trypsin is going to cut after those positively charged residues, we know that there's something that comes after them. Therefore, we have kind of an idea that our peptide is going to end with this peptide that's labeled T3. So what are some orientations or organizations that could be true? It could look like this. We could be looking at a peptide that looks like this, T2, T1, T4. It could be that. It could also be T1, T2, T4, T3. It could also be T4, T2, T1, T3. It could be T4, T1, T2, T3. We have a couple of different organizations. We really don't know at this point in time, but what we can conclude is that T3 is at the end. So we know that our peptide ends with DCBHSD. Okay. Now, what's the amino acid that comes before that D right there? Well, it's either an arginine or a lysine. So what we want to do is then go back and use another restriction enzyme or another chemical to answer that question. What follows it? And when we do that, we are going to use the chemical cyanogen bromide. And that chemical is going to give us the following results. Cyanogen bromide is a chemical that is going to cut after methionine. And when cyanogen bromide cuts after methionine, that means we've got two peptides that end with M. Then we also have this C2 peptide that ends with TK-DCVHSD. Oh, DCVHSD. That's what we saw with T3. And whenever we cut this polypeptide with trypsin, we concluded that DCVHSD was the end of our polypeptide. This was our C termini. So what are the amino acids that precede this? Well, we've got KT. From this peptide C2, we learned that TK precedes DCVHSD. So now what we're going to do is we're going to kind of bounce back and forth. And when we bounce back up here, we can say, oh, well, are there any peptides of these four that, or of these three that end with TK? Yes, there is. T4. So now what we can do is we can say, scribble out that stuff right there. We've got MTK, PGC, AILY. What I did was I said, okay, well, I know that the information that I got from the cyanogen bromide cleavage, I can use to reconstruct the peptides from the trypsin cleavage. And so I can know that this is T3, T4. Now I'm left with, is it T1, T2, or is it T2, T1? Well, what I'm going to do is say, all right, well, I made my T3 or T4, T3 peptide. I've got YLIA. Where is that in my cyanogen bromide clips? Oh, well, here it is. It's in C3. So now what are the amino acids that precede this Y? It's K, I, L, A. Great. Now that I've got that, I can go back to my trypsin cleavage and I can say, oh, well, is there a peptide that ends with ALIK? There is. And before that A is an M. And now I can conclude that, oh, booyah, there it is. So if we look at this in terms of the cyanogen bromide cleavage, C2, C3, C1. If we look at it with respect to the trypsin cleavage, it is T2, T1, T4, T3. So we got this bit of information that we just kind of overlapped them and said, okay, well, trypsin tells us this, cyanogen bromide tells us this. When we put them together, we get a little bit more information. Then we get a little bit more information, a little bit more, and we reassemble through that. Now, the questions that I'll ask regarding this sort of material is I'll ask you to reassemble a peptide. I'll ask, will this peptide be cut by trypsin? Will this peptide be cut by chymotrypsin or elastase? Because those are the three peptides that are three proteases that I want you to be familiar with. Ultimately, this is, I think that these are a series of puzzles. I think that it's very helpful to spend time on this because it gives you insight into basically what a protein, what are the consequences of protein being in the presence of this molecule. All right, well, I hope that was helpful. I hope that it was helpful to break this down into a series of different videos. If you have any questions, please let me know. Other than that, have a good one.